16-year-old, and I get excited about some super weird things. What this has looked like for the past 2 years is waking up at 5 a.m. every day to read research papers about CRISPR, and then getting home from school and spending hours talking with scientists around the world working at the intersection of CRISPR and agriculture. So basically, I'm just a massive science nerd. And my goal for the next 12 minutes is to get you guys as excited as I am about the agricultural revolution that we're on the brink of. And I'd like to start with a little challenge. I'm going to show you the largest innovations that have changed how we grow our food in the past 200 years. But I'm going to do this in 45 seconds. So in 1815, canning was invented, which allowed us to make perishable foods suddenly non-perishable. And then a few decades passed, and in 1835, the reaper was invented. And now we had a way to harvest entire fields of crops instead of spending weeks handpicking each individual one. And then in 1909 came my personal favorite, nitrogen fertilizer, which provided crops with a concentrated array of nutrients in a usable form. Now there have been dozens of these innovations all throughout history, which have created this exponentially increasing graph of global food production. But as the decades began to pass, this innovation in agriculture, it actually started to slow until we're here in 2023, and there hasn't been a major innovation in agriculture since 1952, when chemical pesticides were created. And just by looking at this graph, it doesn't seem like there's a big problem. But if we zoom in from the 1990s to now, projected on to 2050, you can see that the amount of food that we're producing is nowhere near the amount of food that we need to be producing to feed everyone on the planet. And this is a massive problem. And it makes me think of a guy named Thomas Malthus. He was an economist, and in 1798, he came up with this theory that our population growth would surpass the growth in agriculture. Basically, we wouldn't be able to produce enough food to feed everyone on the planet, and then there would be a massive famine that would kill a lot of the population. Now, luckily, this didn't happen because he forgot about the superpower of humanity, and that's innovation. If we didn't innovate and create those things like canning and fertilizer, then his math would have checked out. And it's kind of ironic that after spending decades running away from this food crisis that Thomas Malthus was predicting, we are literally staring it right in the face. We are at this point where low innovation in agriculture is meeting a really high need and creating this crisis point, which has caused that graph of global food production to begin to level off instead of continuing to increase like we need it to. Now, by high need, I mean that there's a super large demand for food. The World Bank estimates that by 2050, we will require 75% more food to feed everyone on the planet. And we will need to produce this increase in food production on a warmer planet with more extreme weather events and fewer resources. So this makes me wonder, how are we going to produce enough food to feed everyone on the planet? Most people say that the answer to this question is to use more fertilizers, add more pesticides, or increase irrigation water. But the truth is that these are all band-aid solutions. They don't actually solve the real problem. That the way that we are producing food, it's fundamentally flawed. To solve this, we need to change how we think about agriculture. And the solution, in my opinion, is figuring out how to do more with less. The only way that we can produce 75% more food is to genetically engineer the crops that we grow to produce these larger yields, but with less resources. Now, let me address a question that a lot of you might be thinking. What does this 16-year-old know about anything, let alone gene editing crops? Well, for the past 2 years, I've been working on creating this supercrop that I'm talking about. I have proposed an idea for a transgenic strand of corn targeted for Malawi, sub-Saharan Africa, that produces 5.5 times larger yields with 5 new target genes. Now you might be thinking, corn, this is cool, but why? And it's because corn is super important. It feeds over a third of the global population, but it's also super inefficient. It takes 127 gallons of water to produce 1 head of corn. And it takes 85 pounds of fertilizer to also produce that 1 head of corn. This is ludicrous. So what I'm trying to do is take this resource-heavy, this hungry plant, and turn it into a plant that can do a lot more with a lot less. And I'm doing this by targeting and identifying the most effective genes in the entire maize genome. Now, before I explain my proposal, let's take a few steps back and clear up any misconceptions that you may have about a GMO. So this word gene editing is just a fancy umbrella term for a bunch of different sciences that give us the ability to change an organism's DNA. And we've been creating GMOs for the past 10,000 years. Started out just by crossbreeding two plants together, and then in the early 2000s, we had technologies like talons and ZFNs, but they were inaccurate. And when a gene was edited, it would cause an unintentional change somewhere down the genome, which is what we call an off-target effect. And I found it so exciting because now we have extremely precise gene editing technologies, like CRISPR, that can edit DNA right down to the individual base pair with basically no off-target effects. So CRISPR is this awesome tool, and we've been using it in healthcare to cure things like sickle cell disease and cystic fibrosis. But how do we use this tool in crops? And it's this application part of CRISPR in agriculture that is the final piece in this entire jigsaw puzzle, and that's gene sequencing and gene mapping. So gene sequencing is finding the order of A's, C's, T's, and G's in an organism's DNA, and then gene mapping is finding the correlation between a specific gene and then its function. And now with companies like Illumina and improved AI and machine learning, we've been able to sequence and map the genome of all major crops to a level of detail that has really never before been possible. And it's because of improvements in these three technologies, gene sequencing, gene mapping, and CRISPR, we can engineer any crops to produce larger yields. So let's say you want to create a strand of corn that produces 10 times larger yields. So to do this, there are a few different levers that you can pull. You can make the plant resistant to pests, or increase the temperature range that the plant can survive at, or you can decrease the amount of water that the plant needs to survive. And then it's all of these levers that, when smushed together into the same plant, then produce these larger yields. And this is not easy. I'm making it sound easy, but it's not at all. The exact A's, C's, T's, and G's that produce that change must be found and then isolated in the DNA. A guide RNA must be created, a plasmid must be designed, and all of this is crazy hard science. And the thing that just blows my mind is that incredibly smart scientists have figured out how to do this process of creating a GMO really well. So we have the technology and we have the science, and now it's just a matter of finding the most effective genes to target. And that's what I'm working on. For the past 2 years, I've spent hours and hours every day reading research papers about improved crop varieties and newly discovered genes that correlate with these higher yields. I've been talking with the scientists and their researchers doing the crazy hard science. And through my research, I found 5 genes that haven't been used before in transgenic corn. And I've created plasmids of each of these changes in benchling. I have presented this proposal to scientists at the International Maize and Wheat Improvement Center and universities like MIT and Harvard, who say that if the 5 genes that I found were engineered perfectly inside corn, this would increase corn yields by 5.5x. Now, let me give you a bit of context on my proposal. So did you know that when the same seed that we grow here in North America, when that same seed is grown in Malawi, in sub-Saharan Africa, the yields are 5 times lower, which creates this endless poverty loop of really low yields and then really low incomes. So my proposal is trying to solve these problems that keep yields in Malawi so low. Things like local pests and like a super long dry season. So imagine if there was a strand of corn that needed 60% less water, grew at 30% higher temperatures, contained 215% more nutrients, and is resistant to basically all pests and diseases. I mean, that would be pretty cool, right? Well, this is what my proposed strand of corn can do. And for all of you nerds like me in the audience, let me give you a sneak peek into 2 of these genes that I found. So the first is abscisic acid, which is a plant hormone that opens and closes the stomata inside plant leaves. When it opens, it lets sunlight and CO2 into the cell, and then it regulates where these inputs travel through the plant xylem. So my idea is to use CRISPR to hack into the ZMP-LY12 gene, which is the gene that creates abscisic acid, and insert the UQ1 gene, which will cause abscisic acid to keep a layer of the stomata closed and conserve water. Now, just this tiny tweak alone, it decreases the amount of water that corn needs by 60%. And here's another example. Phytic acid. Phytic acid is what we call an anti-nutrient in corn, which means that it prevents the body from absorbing the nutrients in food, which then leads to nutrient deficiencies and malnutrition. The biosynthetic pathway that creates phytic acid is called the MIPS pathway. And my idea is to use CRISPR to perform a triple gene knockout, to effectively cut out the 3 key genes at the start of the MIPS pathway to prevent phytic acid from ever being created, which then increases the total nutrient content of corn by 215%. Now, there are 3 more genes that I've identified in corn. One that has to do with growing time, one that has to do with temperature adaptability, and one that has to do with pest resistance. And there are probably at least another 13 other genes, just like this in corn, that haven't even been discovered yet. Now, if we execute perfectly and all this super hard science works out, we can create a transgenic strand of corn that produces 10 times larger yields with 10 times less resources. And it's not just corn that this works for. There are the same amount of these target genes in all other grains and legumes and rice and wheat and potato and soybean. Now, imagine if every crop we grow was genetically engineered to do more with less. This is the scale of innovation that we need in agriculture to produce 75% more food to feed everyone on the planet, to finally start making real gains at solving food insecurity and malnutrition. And this is the reason why I am so excited about agriculture. Thank you.